is swollen and exploded. And this produced much jubilation in the lab. In truth, this photograph I took of Greg Preston was 2 years after our first report. He was still celebrating. And it launched us in a new direction, which we pursued for 15 years, the study of the cellular aquaporins. And we collaborated actively with scientists around the world. I'll skip through the science quickly, because most of it's published, but I'll tell you a little bit about each of the scientists. So to solve the structure of the protein, and the structure is very important for the understanding of the workings of a molecule. Form always follows function. That was Louis K. Sullivan, the architect. In biology, it's the same thing. So the structure of the aquaporin was solved together with Yoshinori Fujiyoshi in Kyoto and Andreas Engel in Basel. And by image reconstruction, we can see that the water channel on the left panel, and this is viewed from above the cell, is a simple donut-like structure with a hole right in the center. I'll try to point to it. 3 angstroms in diameter, precisely the diameter of an average water molecule. And on the right, we have the water channel now in longitudinal sections. So the top is the outside of the cell, the bottom is the inside of the cell, and there's a single aqueous channel with specific residues which repel other substances. We also quickly found that there were multiple aquaporins in the human genome, some which we call classical or orthodox aquaporins permeated by water alone, and some that are permeated by water plus glycerol, which we term aquaglyceroporins. And I'll tell you a little bit about each, but mostly about the people who did the studies. To localize the Aquaporin-1 in kidney, we worked with Søren Nielsen at the University of Aarhus, who localized it in the kidney tubules, allowing the primary urine to enter the tubules with the water being absorbed across the tubule and into the interstitium. And every day our kidneys will filter about 50 gallons of plasma, the same plasma again and again, but the water is conserved to prevent us from becoming dehydrated. People who have defects in the aquaporin-1 molecule cannot fully concentrate their urine when they're stressed. Shown here in the center is a retired school teacher from France with permission. And if you look at the panel on the right, you see normal people can concentrate their urine about 1,000 milliosmolar. That's about the tonicity of seawater after overnight thirsting. She couldn't concentrate it more than about 400 milliosmolar, so there's a clear deficit. The aquaporin-2 proteins were studied also by our friends in Denmark, and it's localized within the cell and allows, again, water to be finely concentrated. And this is a process which we're all familiar with. Fluid excess, we release large volumes of dilute urine. Fluid insufficiency, we concentrate our urine maximally. We do this every day. So the next time you're on an airliner with a full bladder in the economy section and the seatbelt light goes on, I want you to think of aquaporins 1 and 2. They're the problem. Another member of the family is present in lens. Masato Yasui from Tokyo, from Keio University in Tokyo, solved this. And defects in the aquaporin-0 in lens cause congenital cataracts in children. Aquaporin-4, present in the astroglial end feet surrounding capillaries and brain. Following a brain injury, movement into the brain substance could cause terrific problems in the form of brain edema. And the movement of fluid through the aquaporins is what determines that. In collaborating with a group in Norway, particularly with this young scientist, Mahmoud Amiri, Mohadam, we made some profound insight. Now, you don't have to be from Minnesota to realize that Mahmoud Amiri is not a typical Norwegian name. In fact, it's quite interesting. Mahmoud was a refugee from the Islamic Revolution in Iran. He was living in a refugee camp in Pakistan when he was adopted by the Norwegian Social Services, came to Oslo, and given an opportunity, he became a terrific scientist and a leading European neuroscientist. And if you look at the sections of mouse brain, normal shows what a brain of mouse looks like after a temporary defining brain injury, which you see on the left-hand side, pale staining. This is infarcted and edematous brain. Whereas mutant mice with mutations in the aquaporin-4 pathway are spared this defect. If we could develop medicines, If some of the pharmaceutical companies present here today could do that, it might be a very valid mechanism to prevent or treat brain edema, which is a terrible disease and very common. Aquaporin-1 in secretory glands was cloned by Surbi Raina from New Delhi. She's now a professor at Syracuse University. Briefly, the aquaglyceroporins let water and glycerol through because the pore is slightly larger. I won't go through the details. It's very richly expressed. Aquaglyceroporin-3 is very richly expressed in the basal levels of skin. And this is work done by Johan Ogren from Uppsala, Sweden, when he did a sabbatical in their laboratory. Now, it didn't take long for the beauty industry to get interested in this. A few summers back, I had visitors from the executive office of Christian Dior who came to talk to me about giving a lecture in Paris. And I was a bit suspicious of their motives. In fact, they wanted this lecture to coincide with the announcement of a new beauty product called Hydraction Skin Cream, which they were going to market. Now, I have no financial ties to Christian Dior. They offered some, but they all had a lot of strings attached. And I'm not sure that was the best decision. Mary sometimes reminds me it would be nice to have some Christian Dior connections. But we proceeded with the work. Now, their scientists had identified some small molecules that lead to higher levels of expression of the aquaglyceroporin-3 in sun-exposed skin. So that's scientific proof. The notion that if you use enough of this product, you'll look like the model is sheer speculation, but let the buyer beware. If you look closely, this is a back cover of the Marie Claire Beauty magazine. In French, you see profound hydration, spectacular results, and what's this? The Nobel Prize in Chemistry. I showed this to my mother back in Minnesota. She smiled and said, "Peter, I think you're finally doing something useful." I'm not sure this is the most useful, but it does illustrate that your science is public. When you discover something, you don't own it. It becomes public property, and that's where the value really becomes important. So the aquaglyceroporin-3 protein is also present in red cells, where it allows glycerol to enter red cells, which turns out to be very important in the setting of malaria. The malarial parasites will infect red cells, and to make synthetic